So this disease, although it came last year in Bangladesh, Dr. Borlaug in 1995 wrote to Jesse Dubin, who was a wheat pathologist based in Nepal for a very long time, mentioning about this beast will bite one day. And that's what we have seen last year in Bangladesh. Hopefully, we don't see it occurring anywhere else, but that's something which we have. Before I go into wheat, I would talk to about something which is, before I go into blast, I want to talk about wheat. Why are we concerned about blast? We are concerned about blast because it occurs on wheat, it occurs on rice also, but wheat is one of the most important. So this is grown over more than 220 million hectares, both in developing and developed world. Both in developing and developed world, average productions are around 3 million tons, 3 tons per hectare. And this is because most of the developing world is irrigated and most of the developed world is rain-fed. It is used for human food in the form of bread, noodle, and pasta. As fermented product, also it is used in beer, alcohol, and ethanol production, which can be debated when people still don't have their bellies full with bread. We are still producing ethanol from wheat as well. For construction material as roof thatch, and very often the trend is to use it as both as grain and straw for livestock feed. There is a push to develop wheat varieties for dual purposes. That meaning you'll make a cut, use it as forage and let the plant grow, and then that would be used for the grains. It's not really taken up too much acreages, but we know in some areas there is a push to get this dual purpose wheat to come up. Nutritionally, it provides around 20% of the calorie intake of all humans across the world, and there are different classes of wheat. On ploidy levels, you have tetraploid and hexaploid, which you call also as durum wheat or bread wheat. On growth habits, it is winter, spring, and facultative. Kernel hardiness, it is soft and hard wheat. And kernel color, it is red and white. Okay, the kernel color is more important with respect to blast because in Brazil and most of the South American environments, it is red kernel which is grown, while in Bangladesh it is white. So that was interesting because when this question was coming, where did this blast in Bangladesh come from? And the question was, okay, was it South America or not? We knew that in South America mostly it is red grain which is grown, while in South Asia it is white. So there are some differences, but the fungus is adapting to the changing environment and the changing host as well. So where are the maximum productions happening? Globally, I think China for the last long time has been the number one producers, producing around 120 to 130 million metric tons. China has a big challenge of fusarium head blight, and they are very, very worried about wheat blast. Actually, some of the reports of blast being occurring in China, in northern China, was reported. Well, it was not confirmed, and we knew it was fusarium. But they are so scared that they want to have a very good control, and they have already allocated some money for evaluating the resistance of their varieties for wheat blast. India. India has been between 90 plus minus 5 million metric tons. Russia, usually USA has been the third largest producer, but last year it was Russia. EU, Canada, and Australia are the other major producers. What is interesting for us in this slide is, do we have a pointer somewhere? I'm missing it. Anyway, is India, USA, and Australia. These three countries, which are leading producers, are having parts which are vulnerable to wheat blast production. So both India, at least the eastern part, in US, southeast parts of USA, and in Australia it is northeast part of Australia, where the climatic conditions are similar to what is in South America. So there is a chance of this disease moving there as well. Okay, you guys all are very interested in this figure. This is half of the world's population lives in, and this is also the place where half of the world's grain is produced. Obviously, more of rice is produced than of wheat. Another interesting figure is 75% of the farmers live in Asia, mostly in China and India, but other countries are contributing as well. Also, majority of them are poor farmers. So this is important. Well, this slide previously Christian and Dr. Malakar also indicated about is the same story, 1985, first observed in Parana, then slowly it moved around, and in the last 10 to 15, 20 years, it has pretty much occupied most of the warm regions of South America, but has not really hit the major Argentinian wheat production belt. It was always a concern because whenever the disease occurs, it causes major damage, but it does not occur every year. This is very, very unique of this disease, and this is one of the reasons why we have not been able to develop resistant varieties. How do you screen for it when you are not sure the disease will happen or not? And this was the major justification which Barbara and her colleagues used in Kansas, saying that we do not have field-based screening capabilities. Why wouldn't the government fund a BSL-3 laboratory wherein they could evaluate material in US as well? So now at this moment there are two labs, one in Fort Detrick, Maryland, and in Manhattan, Kansas, where they have BSL-3 level facilities and they can do growth chamber basis evaluations for blast resistance. And they have been collaborating very well in our efforts of screening for resistance. But the major concern happened when the disease was observed in Bangladesh in 2016. Yes, please.
Most of the farmers in South Asia are all smallholding farmers.
Yes.
Okay. And in South America, are they smallholding farmers or are they big farmers? So it's very easy to manage the disease in South America because the number of farmers are less and the challenge is more in this.
See, Brazil is where generally people have Brazil and Argentina as very good. When you close your eye, you think, oh, the Brazilians and the Argentinians, everybody is big. No, that's not the story. In Paraguay and Bolivia, the farmers are really very small. The Brazilian farmers, which were the only ones which were large holdings. All the others are very small. So they have also similar challenges as what you would be expecting in Bangladesh. They're a little bit much better in economic conditions, but similar things. These were all shown by Dr. Malakar. Spike is pleased. One of the concerns which we observe is they look very similar to Fusarium head blight. And coming back, I will show you one problem here. Paraguay, okay, between these countries which have the disease, Brazil has the toughest quarantine measures. Seed coming in, seed going out is big challenge. So we have tried to send material to Brazil. It takes almost 4 to 5 years before data comes back. Paraguay is okay, but the problem in Paraguay is you very often see Fusarium head blight occurring along with the blast. So you do not know what you are evaluating for. Bolivia also has some challenge, but it is similar to Bangladesh. They have spot plots and pericularia, which is wheat blast. So for us also, what I see is if you go early in the season before heading, you are seeing spots on the leaf. You don't get confused with spot blotch. It could be blast also, but it's so complex that very often we do not know what it is. Okay, why is blast very scary? The first reason is you are all here. That's why it is, because it must be scary. That's why every effort is being made to mitigate the effect of this disease. However, this is both seed-borne and airborne. This question was raised in the morning session also. The reason why we were very concerned, we kept on talking about seed-borne, is there will be significant implications of seed movement if it is blast. But the amount of inoculum contributing for an epidemic development, is it because of seed or is it airborne? That's something to be debated. What my understanding is, seed-borne component or seed-borne based inoculum is not the major contributor for an epidemic to occur. It is most likely an airborne inoculum coming and causing disease. But it could be an important factor in initiating or development or introduction of a disease. That's what we have observed in Bangladesh as well. No durable resistant cultivars have been identified. There have been some cultivars which show geographically limited resistance depending upon the pressure. Most of the cultivars grown are susceptible, although in the last 2-3 years, especially Bolivia has released some lines, one or two from Brazil also, which show some level of field resistance. There is also a question, what is resistant is a relative term. For example, X variety is very good in Bolivia, but is bad in Brazil. So the first impression is what is happening. Is it really resistant, or the evaluation was wrong? Or it could also be that the fungus is variable. You know, very often in the morning talks we had, we are still very lucky. In Bangladesh, it is a clonal population. Very low. This is one of the major objectives of this training also. When we will collect all the samples, we will try to process it and see if there is variation occurring. Is it all similar to last year, or there has been some changes? If there has been some changes, we need to know where did this changing are happening. Last year's samplings were only maybe six of these isolates were whole genome sequenced by us, and I think some have been done by the European group, but still it was like the maximum 10 isolates. So that's a very limited number of isolates. This year, in this training program, we would look at a very large number and then see where is the variability. Are we fully able to assess and analyze the variability and see what are the implications of this variability in the long-term management of blast disease. The pathogen population is very diverse, exhibits many pathotypes that could cross-infect. Okay, that's another one. The word cross-infect. Highly debated in a meeting in Nepal. We had a researcher, Professor Tiedemann, who gave a very good case, and at the end he said the paper was rejected. He said it was a very good case, and he scared everybody in the audience, and he said it was rejected. So we were a little bit happy that, okay, it was rejected. But that's a challenge in a new environment. How does the pathogen behave? We have to put all efforts to analyze this thing. This is more significant because in Brazil, in South America, you do not have a rice wheat cropping system. In South Asia, you have, 30 million hectares or 20 million hectares of rice wheat cropping system, and there may be opportunities of mutations happening, and that could be very, very, very disturbing. So this has to be also analyzed. Part of this would be also in this training program. Fungicide schemes are partially effective, that too under medium to low disease pressure, and the pathogen has shown some level of fungicide resistance, especially in the case of strobilurine. Reports have come from Brazil that the fungus has developed resistance to this class of fungicides. In Bangladesh, the 6 isolates which were full genome sequenced were all sensitive to strobilurine, but it's just a matter of time. Couple of years exposing the fungus to this fungicide, it may develop resistance. So we have to make a holistic approach of controlling and managing this pathogen. Obviously, it is not only a challenge for South America and Bangladesh, but other countries which have similar climatic conditions should be very threatened and very alarmed of this disease. And these include countries like, well, Bangladesh got it, India, USA, Ethiopia, Australia. They should be very much interested in the research being conducted in Bangladesh and other places, because this fungus can move into their territory and cause damages. The occurrence of the prediction that this disease may occur in Bangladesh was made in a poster by ETN and others from CIMMYT in 2010, and nobody thought that it would come to Bangladesh. Everybody said, how would it go to Bangladesh? But it happened. This is a slide for South America. The disease does not come every year. Initially, the diseases came, the level was less, then the sporicity was there, but in the last couple of years, we are seeing again situations of disease coming more and more. The situation is getting worse, that it is compounding with Fusarium or Helminthosporium. And both these Fusarium occurrences happens in Jessore, and your spot blotch also happens in Jessore. So when you will breed for resistance for blast, you cannot, you should not, and you will not be successful if you just did blast. It has to be compounded with heat tolerance, early maturity, spot blotch resistance, and Fusarium headline. Now our expectation is to do everything, but Fusarium and blast are very, very challenging diseases. So it will be a very slow process, but we have to all walk this hard way. This is another story of Brazil. The acreage went up, and most of these peaks going down are based on the occurrence of wheat blast. More relevant you will see for Bolivia. Bolivia also imports a lot, maybe more than 50% of their domestic consumption is through imports, and they desperately wanted to increase their wheat production, but blast is a very big thing. So big fluctuations are happening, and that's a factor of a biotic thing happening. They do also have a similar challenge of drought, but I think still for them, Pericularia is the number one, they call as wheat blast. I think Christian has shown this slide as well. The pathogen Magnoporte oryza has been pathotyped into different things. Oryza is specific, this is the rice one. The triticum one is the wheat one, and the lolium pathotypes. This lolium ones have been identified in US to cause disease on the wheat as well, but they have not reached to a level that they can be causing epidemics. Under greenhouse conditions, we have to be a little bit careful about greenhouse result being extrapolated into field. Sometimes you do get funny things in greenhouses. We should be careful. Again, this is a genetic diversities work by Mark Farman. Very easily, they are all similar, but based on the host, they can be easily classified of MOT, MOL, and MOL. So although the pathogen is Magnoporte oryza, it has been classified at genetic level and at host level into different pathotypes. And this was again the slides shown before. Okay, there's a very good story here. Okay, okay, this is B71. This is the closest one to the Bangladeshi isolate. B71 is an isolate identified from Bolivia. Okay, all these are Brazilian. So most of the isolates from Bangladesh were closest to B71. That's a Bolivian isolate. So you could say scientifically our isolates are closest to the Bolivian isolate, but reality is Bolivia has never had any export-import from Bangladesh. So you have to be careful in making your interpretations. Please, as young researchers, don't jump to conclusions, what the figures say. This is an indicator. You say, Bolivia is closest, so these all isolates came from Bolivia. No, that's not. It could also be a factor of sampling. It could also, something which Tim was saying, that you will do georeferencing, right? That is important, because what I think is Bolivia, it could be just on the border of Brazil, and you could say it was Brazilian isolate, we just picked it up from Bolivia. I don't know exactly. So one should, before making final inferences, 2 times check and recheck, and then make it. So it was for us very correct that without specifying which country, we could say it was South American. And the more important thing was it was not coming from rice. I think that was the best part of this disaster occurring, that it is not coming from rice or any native wheat, because if it was, that would have been more disastrous. So I'll be talking about the host resistance in tomorrow's talk, but just briefly, among all the resistant genes which have been identified, I think 8 have been identified. All of them are seedling resistance, and most of them have been used, they have been identified on pathotypes which are not triticum. So not the correct fungus was used. The only one which we know which has not been race designated is the 2NS fragment of coming from Aegilops ventricosa. And this was based on breeders' observation that multiple times the lines which were resistant had this segment. And then they started seeing it consistently that when evaluations were there, the 2NS segment was present. And now we have reached a conclusion that 2NS segment does play a very important role. In the adult plant spike-based resistance, we are not exactly sure how much is the role of this translocation and how much effective it is in seedling versus spike reaction. At this moment, what we have reached is there are two mechanisms of, two types or two mechanisms of resistance, which is seedling-based resistance and adult plant resistance. Why? Because there are conflicting reports, some indicating very good correlation between seedling and spike reaction, and others not that good. That's an indicator that maybe there are different mechanisms happening. If you look at Fusarium head blight, which has a lot of similarities with wheat blast, there are 5 mechanisms of resistance. So I believe it could be that in blast also, at least 2 mechanisms of resistance are there, the seedling-based and the spike or the adult plant-based reaction. So this is another good story which Christian would be probably talking about. He has classified technically 4 races. Race 0 is basically avirulent. Race 1 is virulent on Everest 2NS, resistant. Race 2 is susceptible, resistant to Jaglin 2NS, and race 3, it is all susceptible. What was important is in the early days when this disease was identified, you were seeing more of race 0 and 1. However, in the last 5 years, what we hear is more of 1 and 2 is coming. We are still not seeing the super virulent or the race which will be effective against all the 3 checks. Also, all these things which we are presenting has yet to be explored or confirmed in the longer run, because we have only 3 checks. These checks are not single gene checks. See, these are all 2NS, non-2NS based. Then there is another question, is this is non-2NS, this is non-2NS, the reactions are different. Why aren't they the same? That's a question we need to address. So there are beyond 2NS resistance is also there. And that I will talk tomorrow as well, that in some of the cemid germplasm, there is resistance. Actually, Bari Gom 30, right? That also does not have 2NS. That's very interesting, but you have to also understand that's not immune, that's not 0. It has some level. So that's an indicator of partial resistance happening. We need to combine Barigom with somebody with 2NS and then develop the resistance. So you are slowly pyramiding it out. Okay, that's an important step forward. Okay, this is a slide indicating which areas are vulnerable to wheat blast. This was the map indicating the disease would occur in Bangladesh, West Bengal, Central. This is all the hot and humid areas in India as well, and a little bit in part of Sindh. Bangladesh and Nepal and Bhutan are lucky they don't have any problem. Well, if it comes, it will come. Don't worry. This is the challenge. Is all these maps are there with the assumption the fungus will not move, the fungus will not change. The fungus adopts much faster than we think it does, and very often there are unique opportunities happening if it is coming to a new area with so much acreage for wheat. There will be selection for mutations, and they will adapt to the new environment, and that's more threatening. So there is a preliminary study done by a Bangladeshi colleague in Simit itself, Motaleb, and what he observes is that if there was 50% disease loss due to wheat blast, $1.32 billion would be lost. If he looks at what was last year's loss, which was around 25%, this loss ends up into around $500 million. But with 10% loss also is around $263 million. Bangladesh numbers are quite accurate if you look at it. And the Indian numbers are 244 million. So there will be a significant impact of this disease occurring in the three countries. We are not yet there, but we should be prepared. Based on what happened last year, there was a regional meeting mitigating the threat of wheat blast in Bangladesh and beyond, which was held, unfortunately, in Nepal because of the situation in Bangladesh. And what were the outcomes of this meeting was very interesting. A very holistic approach was needed. Some of this have been addressed. Some of this is part of your training program, basically targeting awareness, surveillance, forecasting, epidemiology, development and establishment of precision phenotyping platform. As I mentioned before, the success which we could have achieved in resistance breeding, which we failed to do, was because we do not have precision phenotyping platform where consistent screening can be done. One of the biggest success of DGGW, or at that time DRRW, was the establishment of precision phenotyping platform. In a matter of 6 years, they were able to nullify, if I can say rightly, the effect, the damage, potential damage of UG99. Very often people say, oh, it was not that big. No, it was. It was just nip in the bud, everything done. And similar effort is needed for wheat blast. That we develop a phenotyping platform where global material can be screened at a very high level of accuracy, consistency, and that could be just not for Bangladesh or India, it has to be for everybody. And that is why the name was Global Precision Phenotyping Platform. And from them only we copied this idea to the blast that we need one in Bolivia and one in Bangladesh. Obviously, the major effort needs to be genetics and breeding for blast resistance. I'm sure this will be much difficult task than even UG99. In UG99, you had a lot of expertise. Guys, you have to understand, in rust is one of the most studied diseases in the world. And when UG99 came up, maybe 1% of the germplasm was resistant. But they had very good expertise. Here, we have both the challenges. We don't even know which genetics, which genes are effective, at what level, and what would be the precision. So there's much more difficult task for wheat blast compared to other diseases. However, we have to walk the tough way. Integrated wheat blast management and seed systems, that's another important component. And the last component, human resources and capacity development for wheat blast management, is very much needed. We should not forget last year farmers had to burn fields. This year I think we are cutting them early and grazing them fodder, but this is not exactly what the farmer also wants. At the end of the day, whatever you do here or learn from here, the only reason when you will be successful is if it goes and helps the farmer. If we do not attain that, we are only doing partial success. Obviously, we all want to be successful, but in this disease, you cannot be successful unless you are collaborating. And at this moment, we have tried to develop collaborations in Bolivia, Mexico, USA, India, Nepal, Bangladesh. Australia has put in some money as well. So we are seeing some positive effects of this disease on the donor interest. And hopefully we would be able to get enough funding and enough collaborations among ourselves to successfully mitigate the effect of this disease. And something which our breeder, Dr. Ravi Singh, passed in Nepal. Major losses to wheat blasts in South Asia and beyond can be avoided if an integrated control strategy is implemented urgently, was what was he emphasizing. And that should be it. If you guys have any questions, I'll be very happy to address.